Hello, my name is Alejandro Cañaveral, a dear semester medical student at UPB University. Today, I am here to present the research article entitled CEP 55-3 UTR promotes epithelial mesenchymal transition and enhances tumor density of a blending cancer cells by acting as a ceRNA regulation microRNA 497 5p. Okay, the introduction. Blading cancer is an alteration that occurs when there is a mutation of the urothelial cells that line the bladder. This type of cancer has a high probability of spreading to other parts of the body. That's so. Um, what are the microRNA uh, 497? Are small non-coding RNA molecules that regulate gene expressions at the post-transcriptional level of blading cancer. They can also be used as genetic markers of the same. Okay, the general objective of the authors is we aim to develop potential competition in the origins RNA network related with CP55 in bladder cancer. Okay, the methods. First, RNA extraction and quantity real-time PCR. RNA was extracted from the patient's samples. That RNA was passed to a DNA copy by reverse transcription. PCR was used for the amplifications of the DNA copy. All this with the objective of having a control of the expression of the microRNA. The second method, flow cytometry analysis of apoptosis. Flow cytometry is one of the most popular and versatile applications for studying the apoptosis to make a count of the cells that come to make apoptosis by action of the microRNA. So, luciferase report essay. For microRNA target validated studies, a protein is used that, when it detects transcription activity, will release the luciferase enzyme that will allow it to identify microRNAs. And finally, Western blot, the objective is to monitor antibodies that react with the proteins of interest. In this case, the microRNA, it allows to do this in a very simple way. In monolobelin was visualized using electrochemic luminescence. Okay, now here we go with the results. In the image C, here we are going to evaluate the expression of microRNA 497, CP55, and HMGA2. So, patient 1 has low levels of microRNA and high levels of CP and HMGA. And patient 2 present high levels of microRNA and low levels of CP and HMGA. This shows us the relativity to presence of microRNA. In the image F, in this Western blot, we can see that tumorigenesis may also be associated with the presence of CP55. In the imaging K, we observe the relativity in which the microRNAs will be expressed according to the specific expression of a protein, where B-actin works and control both to have a reference. Okay, the imagined age here it means that our expression of the 3 UTR of CP55 or inhibition of micro N 497 5P in the case of age suppress apoptosis as demonstrated by Nexen 5 senin and flow cytomery. In the imagined A and B is a Western blot, and what you looked was the prometastasis effect of CP55 UTR was associated with activation of innate KB signaling and downregulation of microRN 497 5P transcription. In the imagined C, it is an immunofluorescence, and they look an analysis of the expression of cadherin of bimentin after transfection or CP55 UTR. And the nucleus were stained blue. Now, the discussion. I'm in accord with the first author because this is why bladder cancer gene expressions can be suppressed. Also, I am in accord with the second author because it's correct because bladder cancer can become highly invasive. Also, I'm in accord with the last author because that means there is a present in several types of cancer. Now, the conclusions. Personal conclusions of molecular biology plays a very important role in the eradication of chronic disease. That's why is to research and advance in the modification of factors. And finally, it allows us to move towards the future and it research advance helping all people to have better quality of life. Finally, this is my conceptual map. Thank you.